Houston, we have a podcast. Welcome to the official podcast of the NASA Johnson Space Center, episode 432, ISS Results, Stem Cells in Space. I'm Gary Jordan, and I'll be your host today. On this podcast, we bring in the experts, scientists, engineers, and astronauts, all to let you know what's going on in the world of human spaceflight and more. If you're an avid listener of this podcast, you're likely familiar with science aboard the International Space Station. More than 25 years of continuous human presence and hundreds of investigations. Sure, there are laboratories all over the globe, and many have unique capabilities that draw researchers in to submit proposals and advance humanity's understanding of an endless number of subjects. The International Space Station offers the unique capabilities of microgravity and the harsh environment of space. And a number of facilities on board support experiments in biology, earth science, space science, physics, you name it. For stem cell research, this microgravity environment has welcomed some fascinating experiments. A particular investigation called Microgravity Expanded Stem Cells was conducted on station from 2016 to 2017. The study looked at whether the unique space environment could expand human stem cells, which is a needed process for them to be used in various medical therapies. But in space, how does the expansion process change? The principal investigator of this study, Dr. Abba Zubair, co-authored a recently published research paper about the findings of this study. On this episode, we get a chance to speak with the principal investigator about these exciting results. Dr. Zubair chats with us from Jacksonville, Florida, where he is posted as the medical director for transfusion medicine and stem cell therapy at Mayo Clinic's Florida campus. His focus within this field is on regenerative medicine and immunotherapy applications that involve stem cells and other types of cells. In the meantime, let's get to our conversation with Dr. Abba Zubair about his research and the results that have stemmed from the microgravity environment. Pun totally intended. Enjoy.
T-minus 5 seconds and counting. Mark. Mission start. T0. Launch commit lights are correct. There she goes.
Houston, we have a podcast.
Dr. Abba Zubair, thank you so much for coming on Houston. We have a podcast today.
Thank you.
It's great to have you. I know we've been kind of back and forth trying to make this work. I think we're all busy, but you do a lot at the university that you're at, a lot of research, and you're still practicing. You have to work with your patients, and so the fact that we're talking right now this is certainly very special to us. Take us through who is Dr. Abba Zubair, if you don't mind, maybe starting very briefly with where you grew up, and then how you got to the profession of where you are today, including perhaps maybe inspiration for pursuing medical degrees, and getting your pursuing stem cell research.
Absolutely. So thank you again. It's really an honor to be on this show and talking to our audience, which I'm sure they cut across the nation and maybe abroad. So you can tell from my accent, I'm not from New Jersey, but I grew up in Nigeria. This is a country in West Africa. Went to high school, medical school in Nigeria. But I have been fascinated by space. In the developing world, electricity is not always available. So that could be some kind of limitation. But at night, the sky becomes alive. The stars are more visible. You look out, you kind of wonder. And I'm sure there are many of your audience like that, whether you're in Nigeria or in Florida, wherever you are, some of us are curious. For as far as I can remember, I'm always fascinated by space and what is out there. And I wanted to be an astronaut. And obviously being in Nigeria, that chances are very limited. My career advisor in high school said, look, Abba, you can do anything, but being an astronaut may not be in your lifetime. So you better rethink and do something, more realistic. So I look around and I see a lot of diseases, a lot of people suffering. And I said, well, I really want to make a difference in that arena. So that's how I end up going to medical school. And in medical school, really, it's really fascinating. And we had a lot of experience, expatriate from around the world. We had medical students from US also coming to our medical school. So I get exposed to foreign medical students and we share ideas. I also have become really fascinated by just knowledge and what is out there that I can do. So at the moment, after medical school, I really want to explore. I want to make a difference. So that journey took me through England, where I did a PhD in cancer immunology. And then right after a PhD, I got a green card through the lottery. So I never thought I would be in the U.S., but I just came with a green card. Never had a regular U.S. visa, but then in the U.S. I started at the University of Pennsylvania working as a researcher. But really, my heart is in the care of patients. So even as much as I like the science, I wanted to really also see patients and interact. So I did all the exams. And then I was lucky and opening came, I became available in the same institution at the University of Pennsylvania, went into lab medicine and pathology because I've done a lot of lab work. And I figured that would be a field that would bridge my basic research experience and patient care. But then I end up really loving transfusion medicine, which is really the part of lab medicine that is very clinical. We see patients, we do procedures and stuff like that. So that really attracted me. And one of my clinical roles is to collect stem cells for bone marrow transplant. We collected by apheresis, a device that specialized for doing that. We also process the stem cell in our lab, either from bone marrow aspirate in the OR or by apheresis. We also receive cord blood. So I see the value of stem cell and how it is helping people recover after they have blood cancer. But I have seen patients also that don't have enough stem cells either to donate them for themselves because of their disease. I also see that some patients don't have the luxury of having siblings that can donate for them. So I became very fascinated of finding ways to expand stem cell. But the stem cell is like a seed. They limit their numbers. If you have too much seed, then it becomes like cancer. So when you try to grow stem cell, they always keep their number intact. So I then start thinking out of the box and link my dream of space and say, well, how about if we take out gravity, maybe it will affect the biology of stem cell and make them to grow. And so that's how I end up, studying stem cell in space.
Beautiful story, Dr. Zubair, because this is exactly what we're going to be talking about in detail, is going through, of course, the research that you did in space. And what is exciting is I know you worked with colleagues to publish a paper on some of the research that you have done, and I certainly want to get to that. To sort of ease our audience into this subject and selfishly to ease me into the subject, it's certainly complicated, right? Is stem cell therapy. One of the things you talked about is the clinical aspect of it is this transfusion medicine, and something that may be a little bit foreign to certainly myself, but our audience as well. Take us through exactly what this is and what the benefits are. What sorts of patients would you treat with transfusion medicine?
Yeah, so transfusion medicine, I can tell you, is one of those obscure fields that not many, even people in healthcare, may not know it as a specialty, because again, we are hidden in the lab medicine, which is traditionally not a place where patients' care is involved. What is known about transfusion medicine is blood bank. So the transfusion medicine specialists specialize in transfusion, blood transfusion. We oversee blood bank. We make sure that the blood that comes out the blood bank is compatible, is safe, and we also specialize in managing reaction after blood transfusion. So we work with our colleagues that are more in the front line with the patient. When things happen, then they get to call us. The other aspect of transfusion medicine is what we call apheresis, is a procedure using a machine that looks like a dialysis machine, but it's a procedure that we can collect different blood components either separately or together. So we can select just red cell and take it out. We can take out plasma. We can take out granulocyte and all that. These are all components of blood. In addition to that, we can take out stem cells. So the way it works is that patients get treated with a growth factor that stimulates the stem cell to come out. They grow and then come out into circulation. Then we use the apheresis machine to collect it. So we not only collection, we do therapy. So patients that have, let's say, leukemia, and they have a lot of cells that even their blood is so thick it doesn't flow well. While we are the ones that come with our machine and take out those super excess number of white cells. The same thing also, we treat patients with sickle cell disease. These are patients that have abnormal red cells, and under stress, the red cell becomes sickle and clog their blood vessels, so we can take that out. And then there are a number of conditions that involves antibodies that either mostly the patient develop antibodies to cells, so autoimmune conditions, like lupus, or after transplant patient, they start rejecting their organs, so like kidney or heart. So we can wash out the antibodies that are circulating in the blood to reduce the burden on the organ, so to prevent the rejection. There are other treatments also we do, which some people may not know. We can actually wash out cholesterol. So there are patients that medication doesn't work for them, or they have side effects. So there is a special machine that we can just collect the cholesterol, the bad cholesterol, the LDL, and spare the good cholesterol, the HDL. We do also what we call photopheresis. It's a light treatment that helps patients with some kind of blood cancer, or when they have rejection after bone marrow transplant, what we call graft versus host disease. So there are a lot of conditions that we treat, but generally our role is supporting role. So either hematology or neurology call us to treat specific conditions that are known to respond to the kind of treatment we give. So that's why a lot of people don't know what we do, because we tend to be more a supportive role rather than the primary care team.
Yes, doing the actual transfusion, but it sounds like a lot of, what I'm hearing is this, there are so many applications to this, right? You have patients with this, with an issue, and you're thinking, how can transfusion medicine help this, right? And especially with your work with stem cells, this is where you're thinking, how can I make these stem cells into something that is effective for the patients? And I think that's what's unique about stem cells. Let's back it up to that, right? What is, like, how you're using stem cells. This is what we're going to talk about today, is stem cell and stem cell research on station, but just overview, right? So in terms of the transfusion, you're using stem cells. What are they? Where do they come from? How do you source them for the transfusion medicine? Let's talk about stem cells.
Yeah, so stem cells, they're everywhere. In every organ, there are some stem cells. They are there to participate in tissue repair, regeneration, or of damaged tissue, and so on. However, stem cells that are used clinically, mainly they are used in bone marrow transplant in patients with blood cancer, like leukemia, lymphoma, and those stem cells are collected either from the bone marrow, inside the bone, that's the marrow, that's where they live, and they produce blood cells. Um, so, uh, so our role is to collect that cells and prepare it for treatment to give it back to patient. So there are multiple ways of doing that, So it could be we just collect the cells by the apheresis that I was mentioning, or it might be in the OR where we collect it from the posterior iliac crest, so around the hip bone. We do multiple aspirate and collect the cells, and then we come to the lab and prepare it, and there are different ways of preparing that. And that stem cell can be given right after, let's say, a conditioning chemotherapy to the patient, or it can be stored till when the patient is ready to get that. Another important thing for stem cell therapy is really the whole field of regenerative medicine. Uh, these days, stem cells have been used to regenerate muscle or heart muscle, or regenerate nerve after spinal cord injury, with very limited success, but it's a field that is growing rapidly, finding new application of stem cell in regenerative medicine. A lot of it is still experimental, but we are very much involved in it. And the bigger question is, so if you don't have enough stem cell, what are your options? And for many, the options are very limited. They don't have a relative that can donate because the relative has to be a match to the patient, or we can even search for unrelated donors, and that also is very limited for some, especially minorities, because the unrelated donor registry is not as inclusive, because again, minorities are not represented. So the chance of finding somebody who is a match to a minority patient also is relatively smaller, because the registry is not large enough, and also doesn't have equal representation of those minorities. So.
Yeah, and so certainly, getting more patients, getting more diverse patients, certainly would help with the options that you have, right? You mentioned even, if you don't have enough stem cells, so there's a certain quantity aspect to the application of the medicine that you're looking at, and I wonder if that, and hopefully I'm characterizing this correctly. You can guide me in the right direction, but I wonder if this question, how do we get more stem cells? I wonder if this was the question that sort of led you to bridge, solving for new applications. How can we best use stem cells to help our patients? I wonder if that question right there, how do we get more stem cells, if that was the one that led to your space station research?
Yes, absolutely. So I began experimenting and said, okay, I want to grow stem cell in the lab. So if you have a very limited number of cells, can we take your stem cell and grow it in the lab? And that's where the challenge starts. I mean, we have some ideas. We use cocktail of growth factors and cytokine to grow the cells, but the growth is very limited, because the stem cells are inherently designed to keep their numbers. So when they divide, two cells, one daughter cell become identical to the parent, so it kind of stay as stem cell. The other daughter cells start to differentiate, meaning mature and proliferate. So at every given time, you end up having about the same number of stem cells. Now, there are some tricks we can play to increase the number of the stem cells, but it's still very challenging. And that's why I said, can space environment allow us to do that,
And so maybe did you have maybe some understanding of how the microgravity environment could potentially help in this aspect, and maybe you can start research there? Was there something that helped you to even consider that this maybe had some potential?
Yeah, no, so, you know, key thing about space is limited or absence of gravity. There's gravity everywhere, but in space you get what we call microgravity. On Earth, everywhere, everything we do is influenced by gravity. How we look, we wouldn't have been an erect humans like we do, standing on our 2 feet, if we maybe evolved on Jupiter, which has a lot more gravity on it than on Earth. It's just the mechanics wouldn't work. We might look more like a lizard on Jupiter than somebody, because again, the pressure, the gravity, the weight of the body would be so much to be erect. So that is a good understanding that gravity play a role in our physiology, how we look, but it does also affect how our genes turn on or off. So, and that's why the impact of gravity has gone beyond just the looks or the physics of it, but it does affect gene expression as well. And there's some hints before our research saying that maybe simulated gravity may affect how cells grow and proliferate. So building on that, we said, well, can we really evaluate stem cell expansion at the International Space Station, not using simulated microgravity, but using real microgravity environment offered at the ISS. So that was the trigger, really. And we went with this very simple question: can stem cells grow faster in space at the International Space Station compared to growing them in our lab? So, and that's really the main question. Yeah.
Yeah, and I think this is the study, microgravity expanded stem cells. This is the study that you're talking about, and
Yes.
that's how you can clarify maybe what I was going to ask. What is microgravity expansion? I think it's maybe the growth of the stem cells, right? Maybe I don't know if it's growing bigger or in larger quantities, but is that
It's number. We're interested in the number, so we wanted to see if absence of gravity can allow the stem cell to proliferate, to grow without what we call differentiation. So stem cells are very primitive cells. Their main function is to self-renew and give rise to matured cells or differentiated cells. So we wanted to see whether the cells can grow without differentiating, becoming specialized cells in a microgravity environment. So that was the mission, to see, can we expand the stem cell without them being differentiated, which is the problem on Earth. You stimulate them, they grow, but they differentiate. So you end up having a lot of cells, but they are not stem cells, they are matured cells. So that was the question that we wanted to see in that project. And yeah.
That had to be very exciting, right? You're thinking about that childhood dream in Nigeria, looking up at the sky, having that wonder of space, pursuing stem cells, because unfortunately you were not directed to pursue being an astronaut. But of course, it sounds like you found something you were certainly passionate about. Now you can collect the dots. I wonder if you could take us through those early moments of starting to work, with NASA, with the space station program, U.S. National Lab, to take this idea, this concept. I wonder if, these stem cells can grow in larger quantities on the space station, and working through the process of experiment design, flying it up to the space station, and then executing and working with some of the payload flight controllers to see it through.
Yes, absolutely. I can tell you a lot of things happens because of people. And I just want to give a shout out. There's one person who I say he's the catalyst to my involvement with space research and down to the cases or the National Lab and NASA. Larry Harvey is maybe one of your audiences. He's not a biologist, he's a physicist, and very passionate about space. And he reached out to somebody he knew within Mayo and asking if there are researchers that would be interested to do research in space. So, I mean, that person he reached out to send out email to all investigators. Maybe there are 500,000 of us. And apparently, I'm the only one who responded because of my childhood dream. I like space, and I see this as an opportunity to bridge my practice, my work about stem cell and all the research we were doing. Actually, at the time, we were testing out a type of stem cell in as a way to help recovery after stroke. So I said, well, this is a great opportunity. So I linked up with Larry, and Larry Harvey really introduced me to NASA, to the system and how things work. And the rest of it is history. So it's always about connection and networking, and then obviously you have to have the passion and interest in that arena. So and doing research in space is a team science. You cannot do it alone. Why? Because you have to reinvent a lot of things. You need people with engineering background, because the hardware that you would use would not be typical hardware that you find in the lab. And whatever you would be doing has to be space compatible. So one of the requirements actually for our first project is we have to have what is called implementation partner, and BioStar, which is really more engineering, hardware space entity, is affiliated with University of Colorado, and they have a lot of experience supporting research on the International Space Station. So we team up with them, and they developed the hardware, the bioreactor, the chamber that we would use to grow the cells, because you cannot, things float around, so you can't use just regular hardware that you use in the lab. The other thing, just simply feeding the cell. In the lab, we do what we call media exchange. Media exchange in space is very different from media exchange in the lab. Because of the absence of the gravity, when you mix fluid, they don't mix. They just stay side by side with them. So it's not like you add liquid, then they all mix. No, they don't mix very well. So you have to have a mechanism of mixing, removing old media and replacing it with fresh media to feed the cell. Just as an example of how everything is different when you do. So things that you take for granted on us, especially if you have a lab, you do research, and everything will be different. The other thing, you have to design the experiment in such a way that the astronaut would be able to do it. So, I mean, I wish I could go up there and do it myself, but you have to train astronauts, and you have to make your protocol so straightforward such that it can be performed. If you make your experiment so complex, your scoring will go down, because, again, the astronauts, they are so dedicated and they have tons of projects. So it's not that easy to do that. So again, the project was cases or the first one or the one that we got directly from NASA. I think NASA has been really remarkable because they don't not only just give you money, they also assign a project manager, and they help you with logistics and stuff like that. Together with the implementation partner, which really play a key role, and they are the conduit also between our team and the astronauts that are doing the experiment. They actually train the astronaut. We're not the one doing the training. And they also collect our samples when they return and send it back to us. So they are the implementation partners. They are the intermediary between us, the investigation team, and the astronaut that performed the experiment. So it's really a remarkable experience. Another great thing is that a lot of the actual experiment, when they are being conducted, we can watch the astronaut doing the experiment in real time. And to me, is so wonderful to see your experiment being conducted and you are watching it. And actually, we are able to provide feedback or in real time. So this is remarkable, So, and then we have another set of experiment exactly identical to the one that is being conducted at the space station in our lab, so that we replicate exactly what they have done, and that is our control group. And the publication we have is based on comparing the result from our lab and the result of samples we analyze from space to see, do they grow differently? What are the characteristics, and so on.
Unbelievable. Yeah, you're right. It absolutely takes a team. There's so many disciplines, right? And it sounds like really the idea there is you have an idea of what you want to accomplish in space, and this team of people is there to help you be successful. Think about those things that perhaps is not typically common in a lab on Earth for the specific research you do, and help to solve the problem, because ultimately, at the end of the day, you want good research. You want effective research. You want to make sure that what the data that you're collecting is reliable and is going to ultimately tell a good story for you to do the research that you need to do. I think it's fair to say, because one of the things we're going to talk about here is your paper that you published with colleagues, of course, right? You said it takes a team, microgravity expanded stem cells. I think it's fair to say that all the work that you put into the experiment on the space station at the end of the day, it sounds like it was pretty successful.
Yes, absolutely. Yeah, so the paper is, and there are more, we already published them, and there's still more data. Just on the day we received the samples back, a friend of mine was just asking, so what is the result? I said, well, the result would come, and it may take years to get the final result. Why? Because what the astronaut can do is very limited. So they just can feed the media, they can do simple counting, they can take samples, and the real analysis is when the samples get back to our lab. Then we can do a lot of studies at looking at gene expression, looking at what they secrete in the media, in the environment that they grow. So we did cytokine analysis and growth factor. We also wanted to see if the cells have grown well and are they safe. Because the whole idea of this is using them to treat patients. We are not just doing it for just the heck of it or the curiosity, is application to patient. And so the first thing is to even see was there any microbial contamination. So can the experiment be conducted in a sterile, safe way? And as this may sound trivial, but it is complicated as experiment with multiple people and even astronauts that are not part of the research lab. So there are many opportunities of uh, contamination by bacteria or so, or viruses and all that. So one of the first things we did is to evaluate, you know, how sterile the samples when they come back and all that. The second part is really, somebody asked me, so what did you grow? Did you grow aliens back, in the tube? I said, yeah, there's one cell with 10 eyes. I said, no, I'm just kidding. But truly, we look at the safety of the cells in terms of whether they become cancerous. So have they changed? Have they transformed to become tumors? Because you don't want to grow cells that are behaving like cancer, right? So there are tests we can do to evaluate the safety of the cells in terms of how they transform to be malignant. So this is just broad evaluation of feasibility and application of cells, not so much more to look at their numbers and biology, and that's the other part. So simply we wanted to look at have they grown more than the others, compared to the control. The control are identical to the ones that we sent to the space. So we look at the cell count and see are there more stem cells from the space a sample compared to the sample we have from our lab. And then we also look at the chromosomes. Then we also look at genes and what genes are expressed or down regulated. So, genes can be up or down. So all of that is to see how safe and what is the effect of the absence of gravity on the biology of the cells. The other thing we are worried, so there are three things in space, right, that make this environment so unique. One is the absence or little gravity, so what we call microgravity, right? The second is vacuum. Space is a vacuum. Of course, the International Space Station is pressurized, so it is just like one atmosphere. So that aspect we could not assess because there was no vacuum. The cells are not in a vacuum. But the third thing, which is the elephant in the environment, is radiation, right? So, and one of our interests is to see was there any impact on the DNA? Was there a damage of the DNA from radiation? So those are all broad things, and some of it we are still studying. Some is what you find in the paper, the paper that you are referring to. The key thing about all of this is you should never forget the mission. The mission is to see, can we expand stem cell for human application, right? So that is the key. And so the first paper really focused on that. There isn't much discussion about gene expression, how it affects the biology of the cell. We were just looking at safety, sterility, and any risk of transformation. And that was the paper that you have. The other thing is we didn't only send one type of stem cell. We sent actually 3 different types of cells. 2 are known to have stem cell property. The other one is a leukemia cell line. So we've studied hematopoietic stem cell. We have studied mesenchymal stem cell or mesenchymal stromal cells, and then studied leukemia stem cell. So the paper we published focused more on the mesenchymal stromal cells because the motivation for the project was to see if we can use this kind of cells to treat patients with stroke. Because we have an existing clinical trial using mesenchymal stem cell grown in our lab to treat patients with stroke. So we wanted to evaluate whether cells grown in space can be used in the same condition. So we focused on that at the beginning. And the first paper was really, like I said, we were looking at safety, sterility, and any evidence of transformation. So in terms of the results, we found, yes, we can expand mesenchymal stem cell, we call them MSCs, in a sterile way. So it is feasible to really grow them. The second thing is, can we increase the number? Do they grow faster in space compared to Earth? The answer was no. We didn't see significant growth of the cells in space compared to Earth. This is research. We wanted to see increase, but we didn't see it, and that's what it is. However, this is the interesting part. We found the cells, these MSCs, which are primarily used as immunosuppressive agent. So we use it in stroke to control inflammation, because after a stroke, actually what killed the patient, a stroke is a bleed in the head, and the skull is a limited space. So when you get inflammation and swelling and the bleeding, it really put pressure and can kill patients. So we wanted to see whether we can suppress the inflammation, and that was the clinical study we were doing in our hospital here at Mayo. So the ability of the MSC to suppress inflammation is very interesting to us. And what we found is that the cells that are grown in space are more immunosuppressive compared to the cells that have grown in our lab. So we end up not getting more cells in space, but we get more potent cells. So, and that's really the key. You start with one question, you are thinking you want more numbers, but you may get even same number or less number, but the biology or the physiology of the cell can be enhanced in the direction that you want them to, you hope the cells to be. So basically, we find the cells are much more immunosuppressive. Therefore, for the same number of cells, you may get better effects, better control of inflammation. We also did not see any evidence that the cells have transformed. So, because again, when the cells are grown in space, they back, and you want to treat patients, you don't want to give the cells that are already transformed to be malignant and cause cancer. So that was really helpful. We also see that the shielding around the space station prevent any cosmic radiation to damage the DNA of the cell. So we didn't see any evidence of significant DNA damage. So those are the main findings of that first paper. We have subsequently have more papers, one that focus on what genes are actually affected, what are up or down. So again, we still have samples, we're still looking at different things. The study evolved into looking at bone and bone formation or bone loss, because astronauts have this problem. The longer they stay in space, the more their bone become brittle, so that they lose bone mass almost at the rate of like 1% a month. So the longer they stay, it is quite significant. You start with one question, you have multiple questions. The first space flight we did was only one donor. The next, we have had multiple projects and flights. We have more donors. So, and we also study difference between males and females. And we look at aging as well, so there are multiple questions that evolved out of that first study that we did.
And it sounds, there's so many different perspectives that you could have on just what is seemingly from the outside, someone who's perhaps not a researcher like myself, you just look at stem cells and see what happens, but you have to ask the right questions, right? And what's exciting is how promising you said it was, right? Though they're more immunosuppressant, they're more potent, right? So there's something, maybe not in the larger numbers that we were talking about, but more potent. And I love that you were describing how all along the way, you want to see what happens, of course, but you're always thinking, is this something that we can actually apply to, when we were at the very beginning of our talk, when we were first talking, transfusion medicine, ultimately, you want to help the patients, right? How pristine are the samples that you're getting from the space station? Are the astronauts handling it correctly? Is the radiation affecting it and perhaps making it not, could potentially harm it in a way that would make it not effective? But your goal, you're trying to see, hey, maybe there's a way to make that this could be beneficial in the medical field, and that is so incredibly exciting. I guess exciting enough for you to continue your research, right? And to look at it from all of these other perspectives. I think it sounds like you're a fan of the International Space Station, and you're going to use it. You're going to use it all along the way until its retirement, of course.
As you know, it might soon be retiring, but we hope it will be replaced by a bigger, more capable station. Yeah.
And so it sounds like, and that's of course something that we are looking at. But what we're hearing from researchers like yourself is the microgravity environment is highly beneficial, right? There's something that's happening in microgravity that's good for research that can help us to ask some questions and then eventually unlock new questions, like you said, just by doing the research, right? So it sounds like you're finding the continuation of research in space, highly valuable. You're looking at what's next, and it sounds like you want to be a part of it.
Yeah, absolutely. Couldn't be more excited. I get engaged with from high schoolers to college students to get them excited about space because it's a new frontier, and the good news, we have more access to space because you can see with SpaceX and this other, entities, Axiom and Blue Origin, Virgin Atlantic, that a lot more options now, so this is really exciting time. I really could not imagine because for the first space flight, it took us 2 years to get on the rocket to get there. So now they are having flight almost every month. So it's crazy. So which is really great. There are much more options now.
There's more options, there's more opportunity, and I think that also means it's more promising for your ultimate goal of using space as a platform to perhaps generate more potent stem cells for application to medicines that could be applied to patients. That end goal in mind is certainly very, very exciting. Using space as a platform like that, and the application is there's nothing really more noble than that, right? Is helping people. It's what you set out to do back when you were in Nigeria, and you said, sorry, doctor, or sorry, Abu Zubair, you cannot be an astronaut. But you said, well, how can I help people? And this is fantastic what you're doing. I hope it's awesome, and it's such a noble pursuit. It's really, really cool.
Oh, thank you for the opportunity. Thank you for giving your platform to educate the public for what we are doing, and we want more people to be engaged, to support us, and more people to get into the field as well. So couldn't say it more,
You're very good at explaining things in a way that these are complicated subjects. I tried reading your results page, and my goodness, it goes right over your head. But for you, hearing it from you, you're able to present it in such a very understandable way, and that goes such a long way. I really do hope folks are listening to this and perhaps thinking, how can I get involved? You just got to pursue what you're passionate in and find whatever it is for you. For you, Dr. Zubair, it's helping people, right? With this regenerative medicine, with transfusion medicine, with stem cell research, right? You're thinking of you're attacking the problem from a variety of different ways. People are going to get involved and continue to work whatever happens in low Earth orbit. You know what's exciting is here at the agency, we're going even further than that, right? We have efforts to go to the moon, and of course, all along the way, we're collecting research. I don't know if you thought that far ahead. You talked about the benefits of microgravity in low Earth orbit, one of them being the shielding against radiation. Radiation environment maybe is a little bit more harsh on the moon, but I'm certainly, researchers, perhaps in the medical field, maybe yourself, there's other opportunities there, because we're, talk about expanding opportunities, not only in low Earth orbit, we're trying to get to the moon too, and hopefully that provides yet another perspective for researchers to look at this problem from.
Absolutely. I mean, the moon would have its own challenges. It doesn't have an atmosphere. The radiation would be definitely more. The Van Allen belt, that the magnetic field that protects us, go out to the moon is not as much. So the farther away we go, the more radiation becomes an issue, and we need to find a way. And we are already doing that in our lab, looking at learn from cancer to find ways to develop cells that are radiation resistant. In the end, is how can humans be more resistant to radiation? So we're looking at tools that we can re-engineer cells to kind of make them more radiation resistant. So, and that can play a role in treatment, or, if you're on the moon, there's a big solar flare, you can take some medication to kind of be, more prepared, So we're looking at all of that. It's really exciting time.
Oh, the application to humans. But what you're describing sounds like science fiction. It's crazy what you guys are doing. That is really, really important stuff. And Dr. Zubair, that's where I wanted to wrap up, is that idea, and thinking about the exciting future that's ahead when it comes to these kinds of research activities in space, including yours. Thank you for all you've done on the space station and continue to do. And thanks for taking the time to connect with us and describe the results and the applications and everything you're doing. It was an absolute pleasure talking with you, Dr. Zubair. Thank you.
Thank you so much. Thank you. Appreciate it.
Houston, go ahead.
Stop of the space shuttle.
Roger, 0G, and I feel
The shuttle has cleared the tower.
Became a feat for all mankind.
It's actually a huge honor to break a record like this.
Not because they are easy, but because they are hard.
Never Houston, welcome to space.
Hey, thanks for sticking around. I hope you learned something today. Dr. Zubair and his colleagues' published works is just one of nearly 5,000 publications that have been screened and archived since the station began operations. Go to nasa.gov/station results to check out more that have stemmed from the International Space Station research. I'm sorry, I can't help myself. Our full collection of episodes and all the other wonderful NASA podcasts can be found at nasa.gov/podcasts. On social media, we are on the NASA Johnson Space Center pages of Facebook, X, and Instagram. If you have any questions for us or suggestions for future episodes, email us at nasa-houstonpodcast@mail.nasa.gov. This interview was recorded on June 25th, 2026. Our producer is Dane Turner. Audio engineers are Will Flato and Daniel Tohill. And our social media is managed by Leah Cheshire and Kelsey Howren. Houston We Have a Podcast was created and is supervised by me, Gary Jordan. Thanks to Nicole Rose, Destiny Duran, and Christine Giraldo for their help in bringing this episode together. And of course, thanks again to Dr. Abba Zubair for taking the time to come on the show. Give us a rating and feedback on whatever platform you're listening to us on, and tell us what you think of our podcast. We'll be back next week.
3, 2, 1.
This is an official NASA podcast.